Look at your own hand. Right now, inside the cells of your skin, your DNA is taking catastrophic physical damage. The inside of a cell is a violently hostile environment. Every second, your genetic code is being battered by thermal noise, stray chemicals, and ambient UV radiation. For decades, textbook models and early pioneers like Watson and Crick pictured DNA as a rigid architectural blueprint. The reality is that it's a wet, fragile, constantly vibrating structure that is actively tearing itself apart. If your genetic code is absorbing thousands of physical hits every single day, you should simply disintegrate. Staying in one piece requires a system that can outrun these physical hits, a survival mechanism that operates on a hidden layer of subatomic physics. This diagram shows the electron clouds of DNA bases forming a synchronized quantum shield against thermal particles. But zooming into a single hydrogen bond reveals a vulnerability: proton tunneling. Instead of climbing a barrier, a particle occasionally teleports to the other side. If your DNA splits open for routine copying at the exact microsecond a proton is resting on the wrong side, the cell's machinery is fooled. It reads an A as a G, or a C as a T, and it writes down a typo. The exact quantum mechanics that keep the DNA stable in a chaotic cell ensure that the code will slowly, continuously drift. Spontaneous genetic mutations are a direct result of subatomic physics. Once that typo is made, the cell faces an information processing bottleneck. A tiny pool of repair enzymes must locate one incorrect base pair out of billions in a fraction of a second before it's permanent. Initially, repair proteins use a slow, random method to find damage. They hop across the three-dimensional space, bouncing between distant DNA strands. This vector chart tracks the repair proteins binding to DNA. Once they lock on, the 3D search area collapses into a single track. By restricting movement, they stop bouncing and start sliding smoothly along the sequence, scanning at physically impossible speeds. By mathematically collapsing search dimensions, these enzymes overcome the physical limits of three-dimensional space to process data fast enough to keep you alive. A double-strand break is a structural catastrophe. The DNA molecule severs completely, leaving two loose ends to drift through the cell until the genetic information is lost. This structural failure triggers an emergency first responder, a sensor protein called PARP1. When PARP1 detects a severed strand, it triggers liquid phase separation. A localized droplet instantly encapsulates the break, freezing the broken DNA ends in place. This liquid droplet creates a protected physical zone. It acts as an anchor that holds the strands together so that secondary molecular repair machines can assemble and relink the code. Without the ability to trigger these localized liquid anchors, any total break in the DNA structure would result in immediate fatal data loss. Look back at your hand. In the 5 minutes you've been watching this video, billions of protons have tunneled through your hydrogen bonds. Proteins have collapsed their search dimensions trillions of times, and liquid anchors have sealed countless fractures. In archival images like this cancer research exhibit from the early 1900s, genetic study was limited to macroscopic specimen jars and paper charts. Today, we have finally moved from simply observing this damage to actively manipulating the subatomic mechanics behind it. Engineered cancer therapies now specifically impair the PARP1 superglue, locking it onto the break site. This creates a physical roadblock that the cell cannot clear. When a cancer cell attempts to replicate, it hits that roadblock and triggers its own self-destruct sequence. We are effectively defeating the disease by using its own quantum machinery against it. To see exactly how these targeted inhibitors are engineered at the molecular level, click the next video in our Code of Life series right here.